It will be a brief one actually. We are very much thankful to Professor Budhaditya sir for accepting our invitation amidst his busy schedule. So thank you, sir, for being with us, and we are very keen to hear your expert lecture. I think DBT Star College scheme is all about the interdisciplinarity, maintaining the same, and keeping this, we are organizing this invited lecture with departments of zoology, botany, and chemistry, and students will be highly benefited with such eminent person with you, sir. So, with your kind permission, we can go for the technical session. So,
Thank you. Thank
please.
you so much, ma'am. So now I would like to request Dr. Shumana Datto to please introduce our today's speaker. Sumana,
Thank you,
madam.
Sandipan sir.
Yes.
Good evening, everyone. It is my great pleasure to introduce our distinguished speaker today, Dr. Budhaditya Mukherjee. Dr. Mukherjee completed his PhD from CSIR, Indian Institute of Chemical Biology, Kolkata in 2014. His doctoral research focused on drug-resistant leishmaniasis, particularly how drug-resistant Leishmania parasites manipulate their host, leading to varied disease outcomes. Following his PhD, Dr. Mukherjee joined the Johns Hopkins Bloomberg School of Public Health, USA, for a short postdoctoral tenure. Soon after, he was awarded the prestigious EMBO postdoctoral fellowship and joined the University of Geneva, Switzerland, where he worked from 2015 to 2019. During this time, his research expanded to epicomplexion parasites, including Plasmodium and Toxoplasma. In June 2019, Dr. Mukherjee returned to India and joined IIT Kharagpur as an assistant professor in the School of Medical Science and Technology. His research has consistently focused on immunological and cellular mechanisms that enable intracellular pathogens to survive within the host hostile environment. Today, we are honored to have him with us to share his valuable insights on human diseases and therapeutics, a lifelong quest, role of preferential nutritional acquisition in favoring the development of drug resistance in Leishmania. Please join me in welcoming Dr. Budhaditya Mukherjee.
Thank you. Thank you so much, madam. So, sir, we can go to the technical session. We can start the presentation. Sir.
Okay. Yeah. Thank you for such a nice introduction. So let me start by sharing my screen, and I can switch off my video if that's okay.
Yeah, it's okay. So we can.
Okay, so I guess my screen is visible to you all.
Yes, it's visible, sir. Can you go to the next slide? Yeah, it's moving also. It's moving also. Thank you.
Okay. So as I have been introduced already, I have worked throughout my life on intracellular pathogen. So basically, probably this audience will be knowing protozoan parasite. So basically something that invade us or any other organism and then replicate within that organism for its own survival. So it is basically understandable that this parasites or these pathogens will require nutrients for their division, for their replication, and for their survival. And it has been a long process. So basically, you see that Leishmania parasites have been here for a very long time, and still the parasites is persisting in the nature, in the natural clinical infection. You can see that this is happening all over the world or parts of the world in pockets from some pathogen to pathogen. This varies. This basically means that they have adapted, they have learned new techniques, and also they have outcome the immunological or drug pressure that we have applied on them. So this beautiful picture, if you can see it here, this red color is a murine macrophage cell, and this green color, this green tails here are the parasites which are infecting this macrophage cell. So you see that they live within the host cell. So basically, this is the host cell, and the parasite replicate within it. So today, what I'm going to talk about is that how they have learned to acquire nutrition from their hosts. Particularly, I will be talking about lipids. I will describe to you why we are focusing, why looking into the lipids is important, and what is the outcome of this ability to adapt to take more of the host nutrients. What have it resulted in the parasite? So briefly, you might already know this, but briefly, Leishmania is a digenetic parasite, meaning it has an insect vector, a sandfly, within which it can replicate. And when this infected sandfly basically bite a human, the parasites are dropped in skin, but it does not live within the skin. So once it gets dropped within the skin, it will go to the liver and spleen, and it will replicate there. So all I want you to remember for this parasite is that they are two forms of it. One is the promastigote, which is this flagellated form, and one is the amastigote, which lives within the intracellular macrophages of the liver and spleen, like the small rounded particles you see here. And eventually, when they burden the cell enough with the amastigote load, they will eventually open the cell, and then when a sandfly bite, it will again take this parasite. So there are different forms of leishmaniasis, but we work on the visceral form, which infects in the liver and the spleen. And this is fatal if left untreated. This is how a patient infected with visceral leishmaniasis look like. This is a bone marrow aspirate. And this small red dots you see here is the nucleus of the parasite living within the macrophages. So why we wanted to study this disease? Well, this disease has been restricted in different parts of the world. The disease burden has come down with the introduction of the drug treatment. But the problem still persists because we have seen, like before, we have seen in 2009, there was a drop in disease burden, and then there was again a local epidemic. And India is having major hotspot for this disease, particularly in the Bihar, MP, some parts of West Bengal and Jharkhand. You have the cases or asymptomatic infection or another form of leishmaniasis like PKD, which has come up, but I will not talk about it. Anyway, so although this disease can be treated with the drug, however, still there is significant disease-related impairability and economic burden on the patients, and this normally happens to poor population, economically backward population. So normally there is a chance that they will not go to the clinics and the disease burden will go on increasing. So if you go into the history, Kolkata have a very good connection with the Leishmaniasis or visceral leishmaniasis, because the way how the parasite was discovered was that a patient or a soldier from the British colonial time at that point got infected with some fever in Dum Dum, and then he went back to London, and there was the first time the Leishman Donovan bodies was discovered, and from that we came to the name of the Leishmania donovani. And for the treatment of the disease, if we wanted to look into the treatment perspective, so we must remember this man, even Brahmachari. He was the first person who introduced a drug which was like magic for this parasite, because before that there was no treatment and there was each year a lot of death in West Bengal and neighboring areas. So even Brahmachari introduced antimonials in 1921, and the drug worked like magic. So it was clearing a lot of disease rate, and everything was coming down. And this was very potent drug till late 90s. But around the late 90s, the resistance started to develop against this drug, meaning that antimony, this Leishmania clinical isolates, which were started to obtain at that time from the 1990s, were starting to showing no effect against the antimonial drugs. So with the development of this resistance towards a particular drug, new drugs like amphotericin B, miltefosine, which are still in clinical use, came into picture. However, within a short period, like from 2000 to 2020, the resistance to these drugs have been started reported at an alarming rate. So you can understand that there is a problem that resistance is also developing against these drugs. And this is what I was talking about. Although the VL cases have subsided, we have a new manifestation which is called PKDL, post-Kala-azar dermal leishmaniasis, where this visceral form of parasite somehow lives in the skin of the patient or infect the skin of the patient. So in the title, I have mentioned that I will be talking about resistance. So these are the drugs which were used or have been used at some point for the treatment of the disease. And these are the probable mechanism how they act. Like some of the drugs result in killing the parasite by targeting its membrane. And this is a probable scheme how this parasite have developed the resistance against these drugs. So basically making them ineffective. Now, this is not surprising for a parasite like Leishmania because it has a highly flexible genome, meaning that this parasite can have severe mutation in its genetic organization or plasticity. For instance, normally it can duplicate the chromosome or result in a polyploidy. For instance, let's say it has 2 copies of chromosome 6, but under pressure or drug selection, it can have 1 extra copy of the chromosome just to combat the environmental situation or to neutralize the drug. So Leishmania has a high plastic genome, and you can see that it has a capability to develop multiple copies of the chromosome like monosomic, disomic, trisomic. It is possible for this parasite to have this kind of flexibility. So that could be one of the major reasons that make this parasite highly adaptable to the environmental stress it faces, and probably that's why we have seen the drug resistance appearing in these parasites more frequently. Now, let's look on what we are going to talk about. Now, as I have introduced to you the resistant parasite, as I have mentioned to you before, that 1920s antimonials were used and they were very successful, but around 1990 resistance started to appear, and around early 2000 there was a rapid surge. One of the most peculiar feature of this resistance, as I am going to talk about the adaptability, is a clinical observation. So at that time, Dr. Sipit Thakur was working on Bihar, and he observed an interesting phenomenon that patients who are infected with antimony resistant parasite as compared to antimony sensitive parasite always have one typical feature. They will show a high parasite load in the sorry.
A background noise is coming. Can you please turn off your mic? Whoever mic is on, please.
Yeah, sorry. So if you compare this antimony resistant and sensitive infection, you see that patients who are infected with antimony resistant parasite always have a high parasite load as compared to the infection with the sensitive counterpart. And this is inevitably linked with a huge dyslipidemia, meaning that patient having antimony resistant parasite infection always show a significant drop in the level of their serum cholesterol, LDL, triglyceride. So how these are two related? And this is the question that we ask. This is particularly important for this parasite because if you look into how this parasite replicate, you will see that this parasite come in contact with the host cell as shown in this picture here, and they will attach on the host cell surface. Then they will invade this host cells and started replicating within this host cell in a membrane-bound vesicle, and this is called a parasitophorous vesicle. So basically, this basically keep them secluded from the host cytoplasm, and then they will divide, and this parasitophorous vesicle will grow bigger and bigger. Now, interestingly, the Leishmania parasite are lipid auxotroph. That is, they don't synthesize their own lipid. However, they need lipid. As you can understand that if they have to replicate, they need lipid. They have to synthesize the daughter membrane, and they have to synthesize the PV membrane, which needs to grow to accommodate this parasite. So basically, they are in need of lipid. However, they don't make lipid. So for the explaining the aggressive infection, like where in the resistant parasite you see an increase in parasite load, you can imagine that they will require to take more lipid from their host, and probably that is why in the patient where there is antimony resistant infection, you see a more severe dyslipidemia compared to the sensitive infection. This was the first thing that struck us, and we wanted to understand this process, how antimony resistant parasite have devised a way to probably take more lipid from their host. So the first thing we look at is the attachment. This is how this Leishmania parasite attach on the peritoneal macrophage. You can see here, this is the cell and this is the parasites which are attached with it. So when we compare the attachment of the resistant and the sensitive parasite, we didn't notice any marked difference in terms of their attachment potential, meaning both of them were attaching with their host more or less equally. Like you can see this tail beating here is attached with the macrophage. However, although their initial attachment seems to be similar, when you monitor the infection at the late time point, you see that as compared to the sensitive parasite infection, which we're saying as LDS, the resistant parasite, there is a more load or amestical load. So basically, this means is that this resistant parasite must have proliferated more than the sensitive counterpart to have this many number of amesticles. Now, as I already mentioned, that these are lipid auxotroph. To have more number of daughter amesticle, they will need more membrane synthesis, and inevitably they will need to have more lipids, and this will be coming from the host. So what are the possibilities? So the possibility here is that if you look into this, that here in case of the sensitive parasite, they have a small PV as compared to the large PV of the resistant parasite. As the resistant parasite replicate more and more, they need more and more lipid from their host. However, they cannot synthesize these lipids and they need to get this lipid from the host. So how is this possible? Now, as I have mentioned, that there are certain ways that parasite can take this lipid. For instance, when this parasite attached on the macrophage membrane, which have cholesterol, it can take the cholesterol from this membrane into the parasite. So the first thing we wanted to test is whether this is the case. What we did is we leveled the cholesterol in the membrane. In the screen, as you can see, NBD is a conjugate which we can use to level the cholesterol in the membrane, and then we perform the infection with antimony resistant or antimony sensitive parasite, and then after they have invaded, we collect the parasite by lysing the host cell and quantify how much NBD cholesterol this parasite have taken. However, we observe that in compared to the sensitive, the resistant parasite have not taken much cholesterol from the membrane, meaning during the process of the entry, there is not much difference in terms of the taking up of the host cholesterol. This is just a confocal image representation of the infection and taking cholesterol from the membrane of the host. Now, interestingly, there is other way that this parasite can take cholesterol. So, it is possible by a mechanism which is called a fluid phase endocytosis. So what is a fluid phase endocytosis? To compare that, we just look into the LDL uptake. LDL is the low density lipoprotein, and what we did is we put this LDL, which is in red fluorescence, they are tagged with fluorescence, into the medium where the parasite will replicate. And then we perform the infection to see how much LDL is entering into the sensitive infected macrophages as compared to the resistant infected macrophages. And we have labeled the parasite with a GFP, they are GFP expressing parasites, so you can see the infection. So these green dots represent the parasite and the pink color things represent the LDL. As you can see here, as compared to the sensitive macrophages, the resistant parasite infected macrophages have more LDL in them. We try to monitor this in the live macrophage. As you can see here, as the parasite infection is progressing, you see more and more green color, meaning the parasite is replicating, and you see that the red color is coming, meaning that these macrophages are taking in more and more LDL as the infection process. Now, LDL can be taken up by two processes, which can be a receptor-dependent process or a receptor-independent fluid phase endocytosis. If you understand it like this, that either LDL can bind to a receptor on the macrophage surface and then enter the cell, or it can just pass through the membrane, through the curvature of the membrane, like a diffusion process. And we monitor that if you compare that whether LDL receptor has any effect. So there are some techniques with which we can get rid of the LDL receptor. And we see that knocking out LDL receptor does not affect this process. However, if you block the fluid phase endocytosis, which is a membrane cytoskeleton dependent process regulated by a protein called cofilin, you see that in case of the resistant parasite infection, this cofilin, which is a cytoskeleton protein, which modulate the fluid phase endocytosis, you see this more distributed on the surface of the membrane as compared to the LDS infection or the sensitive parasite infection, which is mostly in the nucleated center of the nucleus, meaning that cofilin have moved towards the surface and probably they are promoting fluid phase endocytosis. So we can monitor this easily because there is specific inhibitor which can inhibit cofilin and block the fluid phase endocytosis. And if you compare this to live imaging uptake of the LDL receptor, LDL, in case of the resistant parasite infection, you can see in the left here, when there is no latranquidine A, which is an inhibitor of fluid phase endocytosis, you see that parasites are replicating, and with that, they are taking in more and more LDL. However, if you look when you have applied latranquidine A as an inhibitor of fluid phase endocytosis, neither you see the parasite replication, and although you see some lipid droplets here and there, you really don't see them accumulating in the cell, suggesting that this resistant parasite are using fluid phase endocytosis as a way to take up the LDL. Now we have few things clear. We know that this parasite are taking up LDL to support their aggressive division or aggressive proliferation within their hosts, and this LDL is taken in the form of fluid phase endocytosis. It is not a receptor dependent process. However, these are two possibilities. There are two possibilities how this can happen. Firstly, LDL can get inside the cell by themselves and the parasite can enter separately. And in order to use this LDL, this parasite containing Parasitophorus vacuole or PV must fuse with the LDL so that they can basically use this LDL. Or what can happen is these two can enter together so that they are in the same space adjacent to each other, and then they can utilize this LDL for their own replication. We wanted to monitor this. So this Parasitophorus vacuole protein is marked by a protein which is called lysosomal protein LAMP1. So LAMP1 normally mark the PV or the PV surface, Parasitophorus vacuole surface is marked by the LAMP1 protein, which is a lysosomal protein coming from the host. Now, if we look into the sensitive parasite and the resistant parasite infection, as you can see here, the lamp one is in green, which should mark the PV or the parasitophorus vacuole, where the parasite is replicating. You put LDL, so you see where the LDL is going or not, and this small nucleus or DAPI is the parasite nucleus. You see here that here you see a co-localization of the LAMP1 and the LDL. So there is some co-localization, but it is not exactly coming to the parasite. However, for the resistant parasite, you see that there is a complete march of this signal, meaning that they are more actively going to the LDL. This LDL are more actively going into the LDR PV. Again, suggesting that this LDL vesicles probably entering independently, but then they are fusing with the LDR PV or the resistant parasite PV with much higher proficiency. So we performed a super resolution microscopy. So this gives you resolution at the nanometer scale level, and you can see here that this PV, which is marked by the LAMP1, this red LDL and the parasite nucleus is being surrounded or taken up by this red LDL, suggesting that this resistant parasite are actually fusing with LDL more efficiently than their sensitive counterparts. Now, only fusing with the LDL is not enough because the parasite also need to use it. Now, normally what happens in a normal situation is that whenever LDL fused with any vesicle, there are some transporter protein on the surface of this vesicle. For example, INPC1. So what happens? This protein normally maintain the cholesterol cholesterol homeostasis in the cell. Basically, your membrane is continuously losing cholesterol, and the endoplasmic reticulum is producing more and more cholesterol in the vesicles, and these are taken up by NPC1, which are then replenished in the membrane so that your membrane architecture is maintained continuously. So, for the resistant parasite to utilize this LDL, have to regulate this release of LDL. So basically this LDL must not get out from this vesicle and go back to the membrane. And we try to look into that. So basically NPC1 is marked in red here. And you can see that there are two adjacent cells here. One for the sensitive parasite and one for the resistant parasite. In case of the sensitive parasite, you don't see that the LDL level is going, NPC level is going down, as you see in this red signal. The parasite containing macrophages have this NPC signal. However, if you look into here, one neighboring cell where you don't have any infection and another adjacent cell where you actually have a huge amount of infection, you see that in case of LDR infection, they actually are downregulating the NPC1, meaning that they wanted to store this lipid within their parasitophorus vacuole so they can utilize it. And actually, we perform a RNA-seq. I am not going into detail of RNA-seq. So basically, you can check whether there is a change in mRNA level of expression which could regulate the protein level expression, and it will give you the entire snapshot picture of the entire cell. And you can see here that along with the NPC1, the de novo cholesterol synthesis pathway in case of the resistant parasite infection, this last column here, remain downregulated. This is not surprising because as this resistant parasite infected host macrophages are taking in more and more cholesterol, they will not like to accumulate too much cholesterol from the host. And so basically what they're doing is they're downregulating NPC1 so that this uptake in LDL does not go back to the membrane, as well as they're suppressing the de novo cholesterol synthesis of the cell, which is regulated by ACRBP2. Normally you have heard about this protein. It's a transcription factor which regulates the transcription or mRNA synthesis of large number of other genes. So normally what happens is ACRBP2 is present in the endoplasmic reticulum, and normally it will go into the nucleus when it is activated, and then it will bind with the promoter of certain genes. For example, here HNGCR, which is the rate limiting enzyme, and LDL receptor, and produce this, increase the transcription of this gene to produce more protein. So when you look and compare actually how much SCRBP2 is going into the nucleus, you see that we have kept the quadrant, and this is the nucleus of the LDR infected macrophage. You see that not only they have decreased the SCRBP2 nucleus, SCRBP2 in the cell, but also in the nucleus you don't see any co-localization of SCRBP2 into the nucleus, meaning that whatever SCRBP2 is left, they are left as a inactivated form. However, in case of the sensitive parasite, you see that this is within the nucleus. So like uninfected macrophage, it is going inside the nucleus. So basically the resistant parasite is taking more LDL from outside, but at the same time switching the SCRBP2 of the infected macrophage to regulate the cholesterol level inside the host. And this can be also visible from other techniques, for example, other biophysical technique. As you know that each lipid could have different type. They could have double bond, they could have single bond, they could be saturated, they could be unsaturated, and based on this, they will give a different signal. And when you measure this signal for the different lipids with the help of a Raman spectroscopy, basically it's a spectroscopy technique, you see that uninfected, LDS infected, and LDR infected cell give a different kind of signal, suggesting that in case of LDR, you have a concentration of particular lipids around the infected amesticle of the Leishmania, which can also be seen by confocal microscopy. This Philippine is something which stain cholesterol, and you can see that they are mostly localized with the resistant parasite, not with the sensitive parasite, where it is dispersed throughout the cell, not something which is concentrated. So we were convinced that the resistant parasite are taking in much more lipid. And further, to convince you that this is actually the case, if you do a live imaging and a 3D projection, you see that this resistant parasite, which take up this lipid, form lipid droplets around this intracellular amesticle. However, in case of the sensitive parasite, the lipid droplets, whatever they are formed, is distinct from the intracellular amesticle. So basically, we can confidently say that the lipid droplets are actually being taken up by the resistant parasite, and it forms a layer of lipid droplet around this intracellular amesticle. Probably this is the source from where they take more cholesterol to synthesize their daughter membrane, synthesize their PV, and sustain their aggressive proliferation. However, this also presents a problem. Problem in the sense that those who understand that lipid biology will know that accumulation of lipid is not beneficial, because when lipid gets accumulated, they always produce an inflammatory response, mainly through IFN gamma, which is a cytokine, and this normally will destroy the cell. So if you try to imagine like this, that the resistant parasite is taking in more and more lipid. So there will be a lot of lipid in this macrophages where the parasite is replicating. However, this also presents a problem because there will be a huge amount of inflammatory response which will eventually destroy the cell. So how it is managing to take in lipid and then replicate rather than being dead by the inflammatory response. So how the inflammatory response against the lipid happens is that whenever the cells take oxidized LDL, which is oxidized, it causes the inflammatory response. However, if it takes the neutral LDL, then it does not cause the inflammatory response. So we try to look into this possibility, and this is actually true because if you stain and try to see how much oxidized LDL is there, you see that whatever lipid is present in case of LDR infection, they are non-oxidized. They does not give this pink color of oxidized LDL. So how this can happen? So normally, oxidized LDL is taken by a scavenger receptor, which is normally present in the liver where these amestigotes replicate. And this scavenger receptor is called emissary 1. And if you compare the expression of the MSR1 in green, in case of the mice, this is a section of the mice liver having the infection. That in case of the LDS infection, you have a lot of MSR1 expression as well as a less amount of neutral lipid. However, in case of LDR infection, you see less amount of MSR1 expression and more of a neutral lipid, meaning that this LDR parasite somehow downregulate the expression of the scavenger receptor and then taking more of a neutral lipid rather than the oxidized lipid. So this basically solves the problem for them. Once they can take the lipid to support their aggressive proliferation, division, produce more number of apelsicle, at the same time, they only take the neutral lipid and not the oxidized form of it so that they are safe from the inflammatory response. And actually, this is also reflected in the RNSET data. The MSR1 is downregulated in the LDR infection. So finally, we wanted to see what is the effect or outcome of this lipid droplet. Why would natural selection prefer the process of this lipid droplet accumulation? Obvious reason is something which is replicating fast compared to something which is replicating slow have always got an advantage. But these have also got other advantages. We see that the lipid droplet here, you can see this green color amastigo. Actually, when we stain this, we see that this lipid droplet actually transfer lipids within this amastigo. And what happens is, these lipid droplets normally surround this amastigo. Now, if you remember, after antimony become resistant for this treatment, there are newer drugs, amphotericin and metafosine was introduced around 2000. And we see that the resistance is developing against this drug. Actually, amphotericin B is an amphiphilic drug, meaning it does not penetrate through the lipid. So we believe, and we actually tested the possibility that this antimony-resistant parasite or those clinical isolates which are showing that primary responsiveness towards antimony due to this lipid droplet accumulation around them, which is needed for their rapid replication, have probably started to showing unresponsiveness against these drugs like amphotericin and metafosine B as compared to the sensitive isolates. Because basically this lipid droplets is not allowing these drugs to penetrate enough to reach and kill the parasite. So this is actually true for several fungal infections where we have seen that this kind of lipid droplet accumulation stop the toxin or trap the toxin from the fungus in order to allow it to survive without getting harmed. And this has been also could be a huge potential for cancer cells where this lipid droplet accumulation and unresponsiveness to particular drug has been noticed. For example, metafosine was originally introduced to treat cancer. And this transmission electron microscopy will convince you. You can see here, this is this amastigote living within the host macrophages. These are the lipid droplet. You see they're closely attached with this amastigote and taking in a lot of lipid droplet, which could probably extend their unresponsiveness to the second line of drugs like amphotericin B. We tried to actually address this because there was a very simple solution to this. You know probably about aspirin. So aspirin is a licensed drug. It is readily available in the market and it has a unique property that it acts as a lipid droplet blocker, basically block the lipid droplet synthesis. So what we did is we treat the infection with aspirin and amphotericin B. The idea is aspirin will solubilize the lipid droplet and then make this resistant parasite more susceptible to amphotericin B. Here is how the experiment was performed. To perform infection, allow the parasite to grow, and then allow them to proliferate in presence of aspirin and amphotericin B, and you see eventually how much amphotericin B is acting on this parasite. As you can see here, that treating with aspirin, it dissolves the lipid droplets. These face-colored droplets are dissolved. Amphotericin B does not dissolve this lipid droplet. And treating with a combination of aspirin and amphotericin B not only dissolve these droplets, but also clear the small nucleus of the amastigote parasites, basically killing the drug resistance parasite. So, sorry, the picture is not here from the lab. Maybe here. So this is our lab. This is the new life science building where our lab is. This work is mostly done by him, Shupratil, and Devolina generated this GFP-RFP parasite. And this project has been funded by ICMR STARS and ACRP. So that will be all. I hope you have an understanding of the infection process, how the nutrients is scavenging, and what is the selective advantage of having these nutrients, how it can how it is promoting a process of resistance to another drug. Thank you.
Thank you so much, sir. It's indeed a very nice presentations, and literally you have covered everything of this pathogen and related topic. So now the session is open for interaction. So I would like to request faculty members, all the students, to please interact with the sir if you have any queries.
I believe that these are mostly bachelor students, so some of the technique might be difficult for them to comprehend, but
yes, exactly.
I try to give them the flavor, like what can happen, what could be the consequence of it, and why we are still facing the problem of drug resistance after so many years, with the selective natural selection giving advantage to a particular form of parasite.
So yeah, but they are all UG students. So few may find it very difficult to understand, but you have done it in a very lucid way. So again, I want to request everyone, if you have any queries regarding anything, please ask him. You will not going to get another chance, actually.
No, you can ask me, no problem.
Yeah, you can. They can ask you later on through email, but this direct interaction is very helpful actually for them.
Yeah.
Yeah, students can also write in the chat box if you want, or they can. So we have actually recorded the lecture, and students will be able to see it on and on. And if any questions comes to their mind, we will be communicating to you. So
just one caution. So most of this data is now in eLife with a revision. So it has not been published yet. Okay, so please don't share it with. The journal has some guidelines.
Yes, sir, definitely, sir. Yes, sir. Definitely, sir. So if there are not any questions, Shondipon sir, then we can just wind up the session. It was, I would say, it is a wonderful session. And the most important is that student got the glimpse of what is going on in advanced research laboratories. Because after the implementation of the NEP, we have internships in national institutes included in our course curriculum, with the students have in their 5th semester, which was previously only for the BTech interns actually. Now we will be having BSc interns after the implementation of the NEP. So with this lecture, I would say students have got a very good glimpse of the microscopy, the use of different advanced research techniques of solving a scientific problem, which is the ultimate essence of this. So many thanks to you, sir, for providing it in a very clear way, student-friendly way, I would say, and very impressive way with such a good presentation. So thank you, sir. So Shondipan sir, I think we can just wind up the session.
We can wind up the session. Thank you so much.
Thank you for having me.
Sir. Thank you, and many thanks to you for giving our students opportunity to listen to you, sir.
Yeah, sure. Thank you.
Thank you, sir.
Okay. Thank you.